The Scepter project, of course, as a lot of you all know, is a very large project. STC are one of many partners that are involved in it, one of several science teams that conduct work across different sectors, and it's not just against pests and pathogens. There's some herbicide work involved in there as well, against weeds. Lots of different partners involved. It's been going on over four or 5 years. The only work I'll talk about here today, of course, is the STC work. I know just about enough of that to be able to take you through that. Other people who are working on soft fruit, on top fruit as well, I believe. And if you do have any questions on any of that other work, I believe we actually have the project leader in the audience. Is that right, Tim? Yes. So I can quickly deflect any hard questions straight over there, or of course to Roma, who's also involved in the work. STC's role in the Scepter project. Glasshouse trials. We have 3 acres of glass at STC to investigate efficacy of selected products on target crop diseases and pests. So a lot of it was almost screening trials, if you like. It's comparing some of these biopesticides to conventionals and to other things like some of the more sort of fatty acid-based, physically acting-based products as well. For pests, we also try to take that a step further and try to investigate the compatibility of selected products with biocontrol releases. This was done back in 2013 in tomato, both with spider mites and with whitefly, I believe. That was actually looking to apply some of the products and then look at them in an integrated regime. We actually applied the products and then introduced biocontrol as well. The advantage, of course, of a lot of these biopesticides is a very short residual toxicity, so you can use them and you can get, even if they're a relatively broad spectrum product, you can get your biocontrol in quite quickly afterwards. Unfortunately, the tomato crop that this crop was done in, that this trial was conducted in, was infected with powdery mildew. It had to be treated with sulfur. It looked like sulfur treatment around about here on the 10th of June actually knocked down a lot of the pests in the control treatment as well as in all of the experimental treatments we're looking at. So it's quite difficult to disentangle what's had the main effect in that trial, whether it was the sulfur treatment or whether it's the actual products that were being tested. So I won't say too much about that. I'll do a little bit more about that one at the actual Scepter conference later on in February. General methods, well, they're all, we touched on earlier that some of these things tend to work extremely well in the lab if you stick them in a petri dish in a very enclosed environment where you've got the humidity, completely set level, temperatures at a set level, day length, etc., light intensity. Things can work very well in the lab. That doesn't mean they're going to translate to the field and work very well there. I know this. I spent probably 10 years doing a lot of the sort of early stage toxicity testing, if you like, doing stuff in petri dishes, looking for products that we can then take on for further testing. And you can get a lot of things to work very well in the lab. They don't necessarily work as well when you take them to the field. So we did all our trials, in fact, all the Scepter trials, commercial scale work. So we're looking at commercial scale glasshouse crops and then managed to industry standards. That, of course, when you're looking at pest work, when I say they manage to industry standards, we, of course, don't treat with pesticides. We don't go in and spray the whole thing with a pesticide. We have to leave the pest there. We actually want the pest there. In fact, we actually put the pest there if we have to. And we do, to some degree, we also have to use things like fleece divides to actually separate rows of crops so that we can have different treatments in different rows and try and limit pest movement between them. It's a way that we try to ensure that we've got individual populations or subpopulations of pests, if you like, that we can actually run our experiments on. They're all replicated studies to make sure we can run statistics on them to prove, to be confident in the results that we get. And they're all conducted according to EPO standards. So the sort of standards that govern how we do these sorts of tests throughout Europe, they're all conducted according to those protocols. Pest diseases and products were selected early on in the project, and as the project was ongoing, if I do make any errors, just jump in and correct me. Through industry consultation and identification of gaps, and that's both existing gaps when the project started and gaps that were foreseen into the future, where we expect products might be withdrawn, where products are at a very high risk of being withdrawn, where they may leave a gap. We looked at biopesticides, we also looked at the sort of physically acting substances, but also novel conventional products as well. And I'll present that data just for completeness so we can actually compare some of the biopesticides to some of these novel conventionals that we also tested. They're all tested against industry standards. So this is the sort of setup we might have in the glasshouse here. This is a pepper trial, and I'm pleased to report we do have some results for pepper, aphids and peppers as part of this presentation. Some encouraging results as well. And what we tend to do is more or less take our commercial scale glasshouse and we'll divide it up into, so we have treatment blocks here. We'll have fleece partitions to try and keep the pests from moving around too much. In between bits, the plots of plants that we're spraying, we'll have guard rows. So we'll have plants that are untreated to act as a buffer, again, to try and limit pest movement. Early on in the project, the experiments were very much of a sort of screening nature of experiments, if you like, lots of products being tested to find out which ones work well, which ones we should take on for further testing. to try and then either get them developed and approved for use at some point in the future. More in-depth testing was conducted, certainly at STC anyway, in the sort of latter half of the project. In terms of pesticides, we screened over 20 products in total, in tomatoes and peppers, and those products comprise botanicals, microbials, physically acting substances, and these novel conventionals. When I say novel conventionals, they're standard conventionals, if you like, but they're perhaps not conventionals that are approved for that crop. So it might be an existing conventional, perhaps, that you want to try out to see if it works in a different crop against a different target pest. It's novel in the respect that it's not currently being used in the crop that we're testing in. Looking at diseases, I am a very long way from being, I'm no pathologist, I'm a very long way from being a pathologist. You will find throughout this talk that the Septa course, and a lot of you, if you've seen Septa presentations before, and I'm sure very frustratingly to many of you, I can't tell you what these products are. And if anybody asks me at the end, I'll run out that door quickly. It's all confidential at present, and we can't release what these actual products codes mean. I'll try to, as we go through, we've got some color coding there. We're actually we've separated things like the biopesticides from the conventionals for you, in some cases the microbials from the physically acting products. But unfortunately, the project only finished its experimental phase late last year, and we're not able to release these products yet, these product names. Diseases, powdery mildew in cucumber. This was a study conducted in 2012, and this is looking at the conventional products here. So we've actually got, this is percentage control compared to the untreated plots. So the standard's actually performed terribly here. You've actually got a negative value. So you've actually, I mean, that's probably no different from the actual control, but the standard hasn't performed well at all. But many of the other conventional products that have been tested here have done very well. And you've got up to and close to 100% control for most of them. For the biologicals, it's more of a mixed bag, if you like. You do have, again, it's far more variable. You've got a few products here, around about half of those products, not much control at all. Extremely large error bars, suggesting that they're very, very variable in their effect. But there are 3 products here, 3 biopesticides. We're actually getting, and certainly for a couple of them, well over that 80%, what we class as control. And the other encouraging thing here is these error bars aren't particularly large, and that means that's not necessarily a very variable response. That's a reliable 80% control. And that can be an issue with some of the biopesticides. So the novel conventionals work well and actually appeared better than the industry standard, and we'll see later on we've found this for a few of the pests as well, the insecticides. Biological products were variable, but several of them, more than 80% control, certainly potentially very useful. Looking at other diseases, unfortunately the results were not as positive for botrytis and cucumber, and I don't actually need a graph for this one. I'm told that this is the typical response with biopesticides, that's the typical response with the conventional product. So no graph needed there and no statistical analysis required. You wouldn't want that, you would want that, wouldn't you? For Pythium, a similar story here. The red bars on this graph, this is the uninoculated control, that one there, and this is the inoculated control. The red bars represent the results with conventional products, the blue bars represent the results with biopesticides. In this case, we've got the higher the value, the worse the product. You've got a plant wilting disease index up there of 0 to 100. So what you're looking for here is lower bars equaling better control or better management, if you like. So in there, you've actually got the biopesticides not performing as well. And that was also the case with Phomopsis as well. I won't dwell on the diseases because I'm an entomologist, so I want to quickly move on to the pests where I actually know what I'm talking about. I'm only going to show you, I haven't got time to go through all the trials that we conducted. Obviously, it's a large project, but I will take you through some of the results we got from 2011 and 2012 onwards after we'd done the initial screening. So we had these initial screening trials, 2011, 2012, and then 2012, 2013, and 2014 were some of the later stage trials. And there are a number of products tested there. And what I've tried to do here, I can't tell you what the products are, but I've tried to, this is an STC coding, these P's, B's, M's, and C's to give you a little bit more information without breaking confidentiality on whether we're looking at botanical products, a B, a microbial product, an M, a physically acting product, something like a fatty acid, for example, a P, or a conventional product, a C. And we're looking at tomato, glasshouse whitefly there, 2-spotted spider mite, peppers, peach potato aphid, thrips, and glasshouse potato aphid as well. Whitefly on tomato. These are the results for adults. What we have down the bottom here on a lot of these slides, you'll see at day 0, that's before we've actually treated them with anything, just to make sure we've got an even pest distribution before we spray. And then we have 6 days after application 1, 6 days after application 2, etc., etc. And you can see here the blue bar there is the control. So 6 days after application 2, all of our products that we've tried there, including our industry standard, but also all of the experimental products, including the biopesticides, are working quite well there, and we're getting control, certainly significant control versus what we're seeing in the untreated plots. And where you see these different letters on things, if there's a bar there that has different letters, it's statistically significantly different. So we can be very confident that this is a repeatable, strong effect. If any of those bars shared a letter, like these ones all share B, they're not different from each other. So even though there may be twice the level of control between these two, it's actually quite variable. They're not really different from each other. For scales, a similar story. All of the products work very well versus the control. So we can say with those data that the standard performed well, more or less as expected, and both novel conventional and botanical products, particularly the ones that we tested, performed well. And they were all comparable, in fact, to the industry standard. So very encouraging for whitefly on tomato. For 2-spotted spider mite on tomato, got a sampling occasion again. This is the data for adults. What you'll actually see here, despite the fact that we've got this very large drop-off in the population here, this data is extremely variable, and we don't actually see any statistical significance. But it certainly suggests that there's something happening here, and that if we repeated this experiment, took it further, ran it again, did it with more replication, this probably would turn out to be significant. And we do have significant results 6 days after application 1 there. There are certainly reductions there, but the data is very variable. It needs to be repeated and confirmed, really, the difference that we're seeing there. The spider mite nymphs, again, a similar story with all of the products that we're testing, including the industry standard, physically acting products, biologicals, microbials, novel conventionals. We're actually seeing a drop-off. It's not always, again, statistically significant. When we work on these quite large commercial glasshouse scales, one of the problems that we have is that we can't have lots of replication, so we do get quite variable data. But we're certainly seeing, quite a significant level of control of the pest population there. And again, reductions there were very close to being significant, but were very variable. Again, standard performed well. Both novel conventional and microbial products also performed well. And even though these differences weren't statistically significant, we're certainly getting reductions there, which suggests there might be products there that will be of value. Peach potato aphid in peppers, which is one that it sounds like a few people will be interested in. And we had some very interesting results here. Blue bars again, this darker blue bar is the control populations where we've only sprayed the crop with water. Quite high levels there. And if we look at some of these green bars, particularly for the botanicals, you'll note the standard's not performing particularly well, pyrethrum. But for some of these blue bars where we've got botanical products, particularly that one there, B130, we're actually getting quite good levels of control 6 days after application 1 and after subsequent applications as well. But it's interesting to note on that one that the standard's doing next to nothing. And again, that pattern's repeated here, but the data is a bit more variable, so we don't see a statistically significant difference. Standard here didn't perform very well at all. That in itself is interesting. B130, however, appears to hold strong potential for peach potato aphid treatment. Thrips and pepper, adults. We didn't see too much before we sprayed after these applications. Here we see some level of control, possibly with B, but it's not actually different from the unsprayed treatment. They share the letter B there, as you can see. Again, this red bar, Calypso, sort of industry standard, it's interesting to note here that we're getting no control at all of adult thrips anyway with the industry standard product. When we look at nymphs, that's also the case. The industry standard's doing next to nothing. When we look at nymphs, we do see, in comparison to adults, where this novel conventional didn't have too much of an effect, we do start seeing a good level of control with this C200 product. So we've got the standard again, not performing very well. We have a novel conventional performing better, although unfortunately biopesticides didn't fare as well in this one. Glasshouse potato aphid in peppers. This was the one trial that I was actually involved in, and it was the one trial where we had a massive infestation, we call it infestation, a massive infestation of parasitoids after we'd started the trial. So what actually happened here is the trial began quite nicely. Before we sprayed, we had a good, nice, even level of infestation across all of our different treatments. 6 days after application 1, when we'd started the experiment, numbers were increasing. That's what we'd like to see in our control, numbers building up. Unfortunately, straight after that, we got high levels of parasitism, parasitoid wasp moving in, the population actually crashed, and it even crashed in the control treatment. So it's difficult to say after about this point. It's difficult to tease out what's happening in terms of treatment, because all of our aphids are being attacked by parasitoids, and they're having an overriding effect on whatever we're trying to do to manipulate the pest population. However, 6 days after application 1, you can see that our conventional is performing very well, as is BO62, one of our biological botanical products. And you can also see here 6 days after application 2, and we've still got data that we can analyse. We're starting to see that both of our botanical products there are actually working quite nicely against glasshouse potato aphid. So in this one, the standard did perform well. Both botanical products performed well. P208 did not perform well, though this may be related to water hardness at STC, and this is something else that needs to be thought about. We've talked about how you require more thought when you're applying some of these lower risk or these biopesticide products. P208 there is a physically acting product, and it's something that could be affected by water hardness. I can't say any more than that. We do have very hard water at STC, and I've since spoken to the manufacturer of this product, where they've trialled it elsewhere, it's worked quite nicely. And it could well be that by putting this into water at STC, the very hard water, the active very quickly precipitates out. It's not something you would pick up when you were spraying the product, when you put it in a spray tank. You would actually have to run if you've got hard water, you'd need to run a quick test beforehand, just something almost in a small vial or a cup or a vase or a beaker. You'd quickly see if that active would precipitate out. And it's something that needs to be considered for some of these products, depending on how they actually function. So in terms of conclusions, biological products performed well against pests, but less well against pathogens. So there were some good results for some of the pathogens that were looked at. Standards did not always perform as we expected, and did not always perform effectively. And this almost reiterates the need to seek alternatives, and in some way justifies or goes part way to justifying the Scepter project itself. We've got standards that are failing. That's not surprising. These standards are based on, if you've got a standard that's based on a single active ingredient, it's not surprising that it fails when you will get resistance building up over time. Several biopesticides have shown promise in trials at STC. Some were effective in multiple crops and against a range of pests. So things like B130 and BO62, this was effective against 3 of our target pests. BO62 effective against 2 of our target pests. What we're trying to do at the moment, certainly for the pest work, and I think through the broader project as well, is to actually build up a picture, now that we have all of this data in, which products are potentially have these multiple effects, which ones are extremely promising on this basis, that they could actually be developed and deployed against multiple target pests and pathogens. No biopesticide proved effective against thrips in later stage trials, though we did find a promising novel conventional product. And potential limitations have been identified in things like water hardness and water chemistry to some extent as well. If you need to think about not just the product that you're putting on, but what you're putting it into to spray it, and of course how you spray it as well. And a lot more thought does have to go into it. It's a more thought-intensive process. And that's it for me. Like I said, I'm a bit of an imposter. I've only been involved in that one experiment. The actual Scepter project is a huge project, as many of you will know. Lots of different partners, and even a lot of the work I've spoke about is not work that I've actually been directly involved in myself. For further information, Scepter, of course, does have a website. We have an HDC news article planned. There are a series of HDC news articles planned, I believe, for different aspects of the Scepter project. I think our pest work is due to be released in spring 2015. And we're hoping to get a sort of open access online journal, particularly on the pest work, so that we can spread the word quite widely through that. And finally, of course, you'll probably also be aware that there's the Scepter Conference, Securing Plant Protection Products for Sustainable Crop Protection. That's taking place on Tuesday, the 24th of February in 2015. And you can actually hear me talk again on the subject. Quite a similar presentation to this one with some similar slides, if you fancied it. That's it from me, I think. Yes, thank you very much.